Hi there, my name is Hind Hashwa, and I am pleased to introduce you to Singlecell RNA-Seq data in GENEVESTIGATOR, a platform containing the world's largest collection of deeply curated transcriptomic data. Single-cell RNA-Seq data are available in GENEVESTIGATOR under data selection, where the studies have been sorted according to the organism and the library preparation. Single-cell RNA-Seq data are available in GENEVESTIGATOR at two levels of resolution, single-cell level and aggregate level. Available for free to all users are two test datasets which allow you to explore these two levels of resolution. SmartSeq libraries with high sequencing depth but a few thousand cells per study on average are featured as one sample equivalent to one cell. The first test dataset is available on the single-cell level and can be found under the organism mouse and the platform Singlecell RNASeq and can be loaded as follows. Droplet-based libraries such as 10X with low sequencing depth but tens to hundreds of thousands of cells per study on average are featured as one sample equivalent to one aggregate, where an aggregate is a group of cells from the same biosample that have shared attributes such as cell type and cell state. The second test dataset is available on the aggregate level and can be found under the organism human and the platform aggregate 10X single-cell RNA-seq and can be loaded as follows. Going back to the single-cell mouse dataset and choosing the right gene selection, if we make use of the condition search tool anatomy, we observe the first anatomical location with two samples corresponding to two individual single cells with the single cell IDs that are visible under the annotation column called single cell ID. Whereas for the other study on the aggregate level, using the same tool but with the right gene selection, we notice the first anatomical location to consist of 8 aggregates corresponding to 187 cells. And in the detailed view, more information on the composition of each aggregate is available under the tooltip. Single-cell RNA-seq data can be analyzed with all the tools that are available in your current license. For example, the human test dataset available at the aggregate level can be used for a differential expression analysis. The study contains over 58,000 cells isolated from healthy individuals and moderate or severe cases of COVID-19 patients. Thanks to the extensive curation of the data by the teams at Nebion, researchers can directly start with their analysis by comparing, for instance, two different cell types or between healthy or diseased cell types, and so on. Here on the left, you can see the different aggregates as this is an aggregate level study. You can make your own group selection based on the different annotation columns that are available here on the right. Or you can make use of the predefined comparisons that have been carefully assigned by our domain-level experts, which can be found in this drop-down menu. If we take a predefined comparison, such as the one observed here, this will allow us to compare between severe and healthy alveolar macrophages using a few clicks and in a matter of seconds. Clearly visible is the added value of the annotated metadata, which equips the user with the necessary information for performing a comparison, whether one is interested in the stimulus, in the sample status, in another attribute such as the cluster-positive biomarker, or any of the others listed here. After running the analysis, we can confirm that many of the top differentially regulated genes have already been described in the original publication, such as CCL2 and CCL7, confirming the reliability and the ease of use of our tools for analysis and visualization of thousands of cells. The two studies used here are available for free to every user for demonstration purposes. Single-cell RNA-Seq studies in GENEVESTIGATOR are available as an extra data subscription for chosen focus areas. For more information on our deeply curated single-cell compendia and to get access, please visit our website or write us an email. Thank you for watching.